Right now, I'm thrilled to be joined by Mohammad Hajj from Bright Biotech. That's a company we've been following with keen interest for more than a year now. Just quickly, Bright Biotech is a molecular farming startup based in the UK that is developing technologies to reduce the cost of producing cultivated meat. And Mohammad is here to tell us how and more. So, Mohammad, welcome to Protein Production Technology.
Hi, Nick. How are you?
I'm fine, thank you very much. Now, tell us about yourself. Where are you from? How did you end up in Manchester and more so working in the field of biotech? Was this something you always wanted to do, or have you kind of fallen into it?
Well, first, thank you very much for having me on this interview. As you mentioned, I'm Mohammed. I'm originally from Lebanon, and I came to Manchester in 2011 to pursue higher education in biotechnology and enterprise. Now, most of the people that come to Manchester, they come either because they're football fans or they're music, fans of the music that comes out from Manchester. But in my case, I came here specifically to join the lab of Anil Day, who's one of the fathers of chloroplast biotechnology. Now, maybe a little bit before I came to Manchester, I did my undergraduate in general biology. And there I came across the different technologies that are used in order to make recombinant proteins. And we've just scratched the surface of plant biotechnology. And this is what triggered my curiosity to want to know more. And chloroplast was a system that I found that there was so much potential to advance on. And yes, as I mentioned, Anil is one of the founders of chloroplast biotechnology, looking into the genetic system of chloroplast to use them as vehicles for recombinant protein expression. So yeah, that was the reason that brought me here to Manchester, in addition to the fact that Manchester was one of the very few universities in the world that were offering a joint program between the life sciences school and the business school, and allowing scientists to bridge the gap between science and business and take enterprise modules to take the research from the lab to the market.
So the company itself, Bright Biotech, was that spawned from the University of Manchester, or is it a completely separate venture?
It's a completely separate venture. So it's the know-how that was developed during my time at the universities, and we've advanced a lot on what was already done at the university and what is already on the public domain.
Mm-hmm. So as the founder of Bright Biotech, I mean, what are some of the biggest challenges that you faced in bringing this technology and the company to fruition?
So it was a long journey. As I mentioned, I've been in Manchester since 2011. The program required me to look at how I can commercialize plant biotechnology or molecular farming. And in this journey, there was a lot of pivoting, trying to understand what is it that the market needs and what are the capabilities of our technologies, what are the shortcomings of our technologies, and how we can address it through the research. So one of the reasons that I didn't mention at the beginning that made me interested in molecular farming is knowing the capability of or the potential of this technology to bring down the cost of making recombinant proteins. And this is something that is very important to me. Coming from Lebanon, we don't have a healthcare system in place. Protein medicine tend to be expensive, and it's available only to the elite, and as is the case in many countries of the world. So the fact that we can bring down the cost of recombinant medicine was very appealing. And the very first project that I engaged in was to make recombinant proteins or vaccine candidates for neglected tropical diseases. And the first business models that I had to commercialize molecular farming was to use the system to make medicines for the people that are incapable to pay for it. Now, it might have been a very ambitious one. So in the journey, there was some pivoting until I landed on an industry that can generate profit to the company at the same time have impact. Because to me, what was the most exciting thing about molecular farming is that you can have a great impact and transform the way proteins are made. And the impact that I always wanted to make was in addressing food security and health security. And this is something that molecular farming allows us to do.
Mm-hmm. So one of the target markets for you, I mentioned it at the start, is cell cultivated meat. So I'm keen to hear your thoughts on where the industry is currently. I mean, there's lots of challenges, lots of pushback in the US, some European countries as well. And sadly, some companies at the forefront seem to be rethinking their strategies at the moment. So what's your assessment of what's going on?
We're strong believers in cultivated meat. Obviously, the timelines that were circulated at the beginning were a bit too optimistic, or they're too ambitious, which is not wrong. It happens in any new or any emerging industry, but I think now everything is becoming more realistic in terms of the timeline. So we're true believers in the cultivated meat. Obviously, it goes through its ups and downs. But it will get there. There are minimal viable products. There are a number of companies that have shown that this technology is possible, but the challenges remain in scaling up this technology, reaching price parity and reaching scalability. So we found that Bright Biotech it provides a technology that addresses one of the major challenges in the cultivated meat sector, and that is the availability and the cost of ingredient proteins that are needed to provide cells with the cues to grow and multiply. So this is one of the challenges. Obviously, there are other challenges in the yields that you can get from larger scale bioreactors, in addition to the regulation. But it seems that, so everyone is working on all of the forefronts in order to advance this technology and get it to a stage where we can actually start realizing the impact that's promising.
Mm-hmm. So in short, you're making growth factors using molecular farming techniques. So what's unique about the process?
So the way we do growth factors is by making use of the chloroplast genetic system. There are a number of advantages that come with using the chloroplast system, and that's why I was very interested in the system from the very beginning. We know that the more common systems are the fermentation-based ones that are reliant on mammalian and microbial cells. They have been used extensively in biopharma. However, they might not be the best models for the food industry because of the infrastructure that is involved in making proteins in bioreactors. Also, there's another factor that comes in, the requirement for high quality water. And also the yields that you can get from these technologies. So you have technological challenges, but also you have environmental impacts that usually is not very much talked about. So with the chloroplast system, we've addressed the issue of yields. The chloroplast, they exist in a very high copy number in plant cells. So we're talking about 10,000 DNA copies. And the way the technology is designed, all of these DNA copies, they take in the foreign gene, giving you a very high number of transcripts, which translate into a very high yield of the protein. In our case, we had yields between 2 to 5 grams per kilogram of fresh leaves. We also address regulatory concerns, which was a major issue for adopting molecular farming in the first generation of molecular farming, mainly because of public concerns of the escape of the foreign gene into the environment and the use of antibiotic-resistant marker genes. So this is something that's addressed in the chloroplast technology. The chloroplasts are maternally inherited, so you don't have to worry about pollen grains containing the foreign gene and escaping into the environment. In addition to that, we've advanced our technology in order to excise antibiotic marker genes. So this is an additional containment advantage. And we shared the other USPs with the higher plant protein production platforms, including the scalability from seeds and the GRAS status, no contamination with animal pathogens and microbial endotoxins. And we have a net zero potential, and this is something very important to talk about.
Yeah, definitely. I mean, could you elaborate on how you target protein expression specifically to chloroplasts?
So this is where the IP or proprietary vectors that we have here at Bright Biotech. Obviously, there are vectors that are communicated through academic research, are already out in the public domain. But one of the cons of chloroplast transformation is the duration it takes in order to generate a transgenic plant. It can take between 9 to 12 months. And this is something that we've advanced here at Bright Biotech. We wanted the process to be shorter. And in order to do that, we've enhanced the design of our vectors. Now, the way we target it, it's without going too technical, but we capitalize on a phenomenon that's present within chloroplast called homologous recombination. So in all standard designs of chloroplast transformation vectors, you have sequences that are compatible with sequences or homologous to sequences that are present within the chloroplast DNA. And by delivering the DNA into the chloroplast, and we do that through accelerating gold particles coated by the DNA into the chloroplast. So they integrate within specific sites within the chloroplast genome through the homologous recombination that is naturally present within chloroplast. So they integrate, and then you use the protein expression machinery of the chloroplast to make the proteins.
You're just at the edge of my technical limits there. So since chloroplasts contain various biomolecules, how does Bright Biotech optimize the downstream processing to efficiently purify that target protein from the plant materials, but while also maintaining integrity, I guess?
Yeah, so downstream processing is actually a challenge in all protein production platforms, and it is the case for plant biotechnology or molecular farming as well. First of all, you have to process large amount of leaf biomass. And then, as you're saying, the leaf biomass, it comes with a lot of host cell proteins. So you need to do some sort of clarification. Now, it depends on how the protein accumulates inside the chloroplast. So we've seen that some of the proteins accumulate in soluble form, which means that you need to separate them from the host cell proteins. And for other proteins, they accumulate similar to what is known as inclusion bodies in microbial systems. This is a faster purification because in one step of purification, you can get 80% purity. But the downside is that you have to do some chemical refolding after that, which can add up to the cost of downstream processing. So in our case, it varies from one protein to another. But definitely, a lot of innovation needs to go into the downstream processing in order to optimize the yield and maximize the conversion of the 2 grams or 5 grams per kilograms of fresh leaves into active products.
When it comes to large-scale expression in the chloroplast, is there any negative impact on the organelle's natural photosynthetic function or overall plant health? How do you monitor those potential negative effects, if there are any?
So there has been reports of high levels of expression of recombinant proteins affecting the growth of plants or affecting the fertility. So you might have like a male sterile plants. We haven't seen it with our proteins or the proteins that we've used to exemplify or do a proof of concept for our technology. Now, the advantage of using chloroplast, so you might see more of these negative impact on the growth of the plants if you introduce them into the nuclear genome. Now, the chloroplast, it's compartmentalized, so you minimize the toxic effects of the recombinant proteins on the health of the plant. So that's why there has been few reports, but it's not something that's common.
Mm-hmm. Now, scaling is something we talk to everybody about. How do you envisage scaling this method of protein production for large-scale commercial applications? And can it actually be cost competitive with other protein production methods? Where are you with scaling, and where do you hope to be in terms of timelines and volumes?
That's a very interesting question, because now we're looking deeply into a life cycle assessment and a techno-economic analysis, and then comparing it with all of the other technologies. But just like from the readings that we've already done, we can already see why molecular farming is the system to go for the future. Obviously, I'm not saying that molecular farming is capable of doing all of the proteins. We will still need the fermentation-based technologies. But then for the ones that molecular farming can do, I think it is going to be the go-to platform. So in terms of scalability, we're looking into open field growth. We're looking into vertical farming. There's a lot of innovation that happened in vertical farming in order to make it more sustainable compared to open field trials, because every approach has its pros and cons. So open fields, obviously, you're bringing the cost like significantly down because you're using traditional farming, which is a lot cheaper than having to operate a very complex fermentation-based facility that requires highly skilled people. But the downside of using open fields is that it's a water-intensive industry. You might need fertilizer, so it's being grown in an open environment, so it might be a bit challenging. Now, in a vertical farming, you will have the energy consumption because you're dependent on LED lights. Now you're bringing down the cost as much as possible with using sustainable. You might be you'll have to be dependent on renewable energy, but also water consumption is significantly reduced compared to open fields because you're recycling the water. So what we're doing here at Bright Biotech is we're exploring all of the approaches that are available to us and then comparing them and trying to find the best approach to scale up our technology. But it's looking like the vertical farming is the winning approach, but the numbers will eventually tell us which one to go for. Even one thing, just you've asked me about comparing the two approaches, fermentation-based and molecular farming. So as I mentioned in the beginning, molecular fermentation-based technologies, they're dependent on high-quality water, which is not the case for molecular farming. They are dependent on non-renewable material, as opposed to molecular farming that only demands light, water, and CO2 in order to drive the expression of the protein. So, so in terms of the upstream processing, definitely molecular farming has a winning case compared to fermentation-based technologies. You're scaling up from seeds, and so even looking at the waste streams with the fermentation-based technologies, the treatment of waste is going to be very costly. As opposed to molecular farming, you have a lot of waste material generated at this point, but then we are thinking of a circular economy in order to make use of all of the secondary metabolites that can be used in other industries, and minimize the environmental impact from a process that makes recombinant proteins.
Mm-hmm. Now, the plants are not natural protein factories for complex animal proteins. So how can you improve the efficiency and yield of target proteins within the plant cells themselves?
So again, it all goes back to the design that we have in place. You can enhance the yield of the protein by playing with the regulatory elements that are present within the vectors. So this is one thing that can enhance the yield within a plant. Now, for everyone knows that animal proteins, they are more complex, so they have post-translational modification. Chloroplasts, they come from a prokaryotic ancestry, so even though they are hosted by a eukaryotic cell, but most of their features are prokaryotic or they are microbial. So sometimes they struggle in making more difficult proteins. But then they do make disulfide bonds. They have their own disulfide isomerase. Sometimes it works brilliantly well, other times it doesn't. But so this is why it's very important also to optimize the downstream processing. There has been, for example, bacteria, they don't do post-translation modification, but then pathways that does it have been engineered into bacteria. So probably in the future, we might be looking at ways where we can enhance the capabilities of chloroplasts of doing post-translation modification. And by doing that, we'll be able to make more complex proteins in our system.
Now, you touched on regulations a little bit earlier. Are there any specific hurdles for chloroplast-derived proteins compared to those produced in other plant compartments? Mm-hmm. Or how are you navigating those regulatory considerations?
So as I mentioned that we addressed regulatory concerns. So one of the major issues was the escape of the foreign gene into the environment and contaminating food chains. So first of all, at Bright Biotech, we're using tobacco as a plant. So here in the UK, it falls within category 1 or lowest risk because tobacco is not grown in the country and the relatives of tobacco are not grown in the country in the open fields. So it's lowest risk as compared, for example, to using a leafy crop like lettuce. which might cause some concern. But as I mentioned, the chloroplasts are maternally inherited, so the pollen grains would not carry the foreign gene. And we developed a process on the same design that we've put together in order to enhance the efficiency of the process. We've also allowed for the removal of antibiotic-resistant marker gene, which is most of the time using all of recombinant protein technologies in order to generate a transgenic line. And sometimes it is necessary to remain in order to keep the gene stable within a genome. So in our case, we excise the antibiotic-resistant marker gene, and it doesn't affect the accumulation of the protein from one generation to another generation. So this is an additional advantage. Now, also in terms of safety, the plants, they are not natural hosts for animal pathogens. So downstream processing, it's a lot facilitated. You don't have to worry about your end product being contaminated with animal pathogens. Also, the same applies for microbial endotoxins. They don't carry any endotoxins. So you have a strong case for potential regulatory clearance, and this is something that we are going to look at next year at Bright Biotech.
Marvelous. So in what ways could your particular strand of this novel foods era integrate with other alternative protein technologies to create a more diverse and robust food system for the future?
So basically, this is what we have at Bright Biotech. So we're integrating molecular farming with the cultivated meat sector, which is heavily dependent on bioreactors at this stage. So we're bringing these two together. We believe that it doesn't make any sense to overwhelm the infrastructure, the fermentation-based infrastructure, because a lot of it is going to be allocated between the biopharma and between the cultivated meat sectors. So why use the fermentation-based technologies just to make one ingredient for the process? So this is where molecular farming comes in. It's just like it's providing, it's enabling the cultivated meat sector without putting any more strain on the existing or maybe non-existing infrastructure, because there isn't like yet an extensive industrial scale infrastructure for cultivated meat.
Onto fundraising. Now, you've enjoyed some success. I think off the top of my head, I think you had a raise of around $3 million in November 22, I think it was. Not too sure. But where are you currently in that adventure? And what advice would you have for other startups not so far ahead in their journeys?
So yeah, our fundraising journey was very exciting. We were in a very good position. We were oversubscribed, and hopefully we'll be able to replicate this for the next funding round. But yeah, so now we're capitalizing on the first fund that we've raised. We're growing the team. We're now 9 people in the company, and we're growing organically. And everyone that's coming into the team is fitting so well and is aligned with the core values that we put for the company. Obviously, we've set up our own laboratory in order to be independent of any third parties, and this is now up and running. We're doing our processes from A to Z, starting from introducing the foreign genes into our plants, to processing the leaf material, and also doing the in-house validation. We have now a runway for another 15 months, but we're going to be actively fundraising starting September. So we're always up to warming up with investors. And we're always excited to show people what we are doing, especially like investors, because to move to the next stage, obviously, we need some support in scaling up our capabilities, and which will allow us to take our products to the market and also expand our product portfolio.
We had a feature on investment in our previous edition, and the majority of the people interviewed for that feature said that they were looking to invest in companies such as yours, the facilitators of the end product rather than the companies producing the end product. So that's going to be good news for you, I hope.
Yes.
Now, looking back, sorry, go on.
Yeah, I was just going to say that that's very interesting to see the shift, because I remember when I first attended, it was industrializing cultivated or cell-based meat and seafood. This is when I first got introduced to the industry, and there was a panel discussion which involved investors. And they were saying they are interested in investing in the end producers rather than investing in the enabling technologies. And it was discouraging, just like to hear that at the beginning of the journey. And yeah, so in the past 2 years, we've seen a shift, and then seen that a lot of investors are now interested in supporting the enabling technologies, which makes complete sense. You need investment to be split between the two, because eventually we are in this business to enable and enable the cultivated meat and allow them to overcome their challenges.
We're based in the UK. Now, what's the stance at the moment of the Food Standards Agency? They seem quite welcoming of food tech generally. So, what's your opinion of the UK market for cultivated meat?
Well, first steps have been taken. My understanding that cultivated meat has been mentioned in a governmental letter or policy. Obviously, so more action needs to be taken. Maybe having a regulatory framework in place and with clear questions, so that the enabling technologies and also the end producers understand what exactly that the regulators are or need to see in order to assess a product. So maybe. We still need to look more into the regulation, but yeah, so it's definitely the cultivated meat is being mentioned. There is Alternative Protein Association here in the UK, which includes a number of companies that support the cultivated meat sector, or they are in the cultivated meat sector. So there are also funds, and there are big syndicates of a number of players in the industry coming together in order to address challenges. So definitely the UK, yeah, so it's in the right direction. And I always say that when we ask for a regulatory framework, is not asking for a fast track one, because at the end of the day, we always need to ensure that the human welfare is well taken care of. So it's not the skipping steps in order to render a product safe for human consumption, but rather having a clear route that people can follow, so that we can take this product to the market.
Have you tasted cultivated meat, Mohammad?
Unfortunately not. So I've been twice in Singapore, but I didn't get the chance.
You couldn't get along to Hoover's?
No, but not exactly now. Yeah, so I know now that they're sending it to Hoover's, so which is it's a great step forward. So even though it's just like 3% of the end product, but that's like a great step to commercialize it at a larger scale.
Yeah. So I think Vow as well, they're now selling it in a restaurant, their Qualia product. So yeah, we will taste it eventually. Now, looking back,
Yes,
would you have done anything differently with how you set the company up and evolved the technology?
Well, obviously, there were a lot of challenges in this journey, but every challenge was a learning experience for me. Coming from a scientific background and entering into the business world, I really enjoyed all of the challenges that I've come across, whether it was in setting up the company, growing the team, putting systems in place in order to honor the values that we've put in the company, which revolve around sustainability, collaboration, being innovative and enterprising, and doing excellent or high quality work. So all of these different business activities, they come with challenges. So would I change anything? No, because if I've changed something, probably it wouldn't have been as rich. The experience wouldn't have been as rich as it's been so far.
Mm-hmm. So in your personal view, what key milestones would signify success for you and Bright Biotech in the next sort of 5 years or so?
So a key milestone would be if Bright Biotech becomes the go-to supplier of growth factors for the cultivated meat sector. Now, obviously, there are other industries that are growth factors could be applied to, like R&D and regenerative medicine, but we really want to enable the cultivated meat. We believe that we've got what it takes to help them move or transition from lab scale to industrial scale with the scalable technology that we have. And the yields that we have and the potential or the cost opportunity that we provide through our technology. So yeah, by capturing or attracting the majority of the players in the cultivated meat sector, so that would be a key milestone for us. But on our side, we have to broaden or expand our product portfolio. So before we get to the stage, we have to do a lot of work on our side. And bring a bigger portfolio of products to the cultivated meat sector.
Well, look, Mohammad, that's all we've got time for. It's been absolutely fascinating talking with you. Thank you for keeping it just within my knowledge limitations. It was technical, but not amazingly technical. I'm sure our readers will love hearing from you. So look, it's been fantastic speaking with you. I'm sure we'll catch up with you again soon, and we can only wish you and your team all the best for the future.
Thank you very much, Nick, for your time, and I really enjoyed this conversation.
Great, thank you.